our today's module is extrinsic factors affecting microbial growth and survival in food. In this module, we will understand how external or environmental factors affecting microbial growth and survival in diverse food environment. The growth of microorganisms in food can be affected by several factors present inside the food or the outside environment where food is kept. These factors play an important role in understanding the growth of microorganisms in food, and by these factors we may know how to keep our food safe for consumption. Such informations are also helpful in designing better conditions for preserving food products for a longer period without losing the nutritional values. In general, extrinsic factors are factors in the surroundings and external to the food that affects both microorganisms and food itself during processing, handling, and storage. Extrinsic factors affecting the growth of microorganisms in food are storage temperature, relative humidity, gases, and activities of other nearby microorganisms. Diverse microorganisms grow over a wide range of temperature. For example, some microorganisms prefer to grow in the cold, that is low temperature, few at room temperature, and other to grow at high temperature. While the growth rate increases with rising temperature, the growth rate tends to decline rapidly after that until the temperature maximum is achieved. Both low and high temperatures may affect the rate of microbial growth. At low temperatures, microbial growth ceases because of slower reaction rate for individual enzymes, and at low temperature there is a decrease in fluidity of the cytoplasmic membrane which affect its transport mechanisms. At high temperatures, there is a denaturing of structural cell components as well as inactivation of heat sensitive enzymes which may occur. Food and food products are exposed to different temperatures from time of production until the time of consumption. The microbial growth is accomplished through enzymatic reaction which is dependent on temperature. Every 10 degrees Celsius rise doubles the catalytic rate of enzyme, and every 10 degrees Celsius decrease reduces it to half. Normally, all microorganisms have a defined temperature range in which they grow. Actually, the temperature range over which a microorganism grows that reflects the temperature at which its enzymes function. So within this range, three cardinal temperatures can be identified: minimum, optimum, and maximum temperatures. Based on temperature requirements for microbial growth, microorganisms may be placed into three groups: psychrophiles, mesophiles, and thermophiles. Psychrophiles are those microorganisms that grow well at or below 7 degrees Celsius and have their optimum between 20 degrees Celsius and 30 degrees Celsius. These organisms grow well at refrigeration temperatures and responsible for spoilage of meat, fish, poultry, eggs, and many other food normally stored at this temperature. For example, the genera belonging Pseudomonas, Enterococcus, and others such as Alcaligens, Schwannella, Corynebacterium, Lactobacillus, Pseudomonas, Psychrobacter, Enterococcus, etc. Mesophiles are those microorganisms that can grow well between 20 degrees Celsius and 45 degrees Celsius with optima between 30 degrees Celsius and 40 degrees Celsius. These type of microorganisms may be found in food stored at refrigeration temperatures. In fact, they do not grow at this temperature but do grow at temperatures falls in the mesophilic range depending on other suitable conditions. It has been observed that Enterococcus faecalis can grow over a range from 0 degrees Celsius and 30 degrees Celsius or above. Thermophiles are able to grow well at and above 45 degrees Celsius with optima between 55 degrees Celsius and 65 degrees Celsius. For example, Bacillus and Clostridium, the genera belonging to this group, are of great interest to the food microbiologist and dairy food technologist in the canning industry. Besides bacteria, interestingly, yeast and molds can grow over a wide range of pH, osmotic pressure, and nutrient content, likewise also able to grow over wide ranges of temperature. Although yeasts can grow over the psychotropic and mesophilic temperature ranges, but usually not within the thermophilic range. Numerous molds can grow at refrigeration temperature, especially some strains of Aspergillus, Cladosporium, and Thamnidium, which may be found growing on eggs, sides of beef, and fruits stored at low temperature. Hence, the temperature of storage is most important parameter that affects the growth of microorganisms and their survival in food. Microbial growth at food surfaces also affected by the humidity of storage environment. If we store food in a dry atmosphere, means that less humidity, microorganisms are less able to grow than if the food is stored in a moist environment where high humidity exists. That is why dry conditions are considered better for storing food than moist conditions. In general, there is a great interaction in between the water activity of food and relative humidity of the storage environment. It is important to note that if the water activity of a food is set at 0.65, it must be stored under conditions of relative humidity that do not allow the food to capture moisture from the air and thereby increase its own surface and subsurface water activity to a point where microbial growth may occur. There is always an equilibrium in between water activity of food and relative humidity of the environment. If, for example, if food with low water activity values are kept in environments of high relative humidity, in such condition, the food pick up or capture moisture until equilibrium has been established. Similarly, food with a high water activity lose moisture when placed in an environment of low relative humidity. Temperature is also affecting the relative humidity of the environment. So, we should be careful while selecting proper storage environments for food. If higher the temperature is higher, then the relative humidity will be lower and vice versa. Aerobic microorganisms require oxygen for their growth. For example, E. coli is a fecal bacterium which grows readily in many food. If food is stored in a reduced oxygen environment, then aerobic bacteria would not able to grow and reproduce. In contrast, there are few microorganisms that can grow in absence of oxygen called anaerobic microorganism. The best example of anaerobic microorganism is Clostridium botulinum, which causes botulism. Microbial growth is affected by gases with two mechanisms called direct effect and indirect effect. Direct effect: some gases such as carbon dioxide, ozone and oxygen directly toxic to certain microorganisms that can inhibit growth and proliferation. Their potential is dependent upon the chemical and physical properties of the gas and its interaction with an aqueous and lipid phases of the food. And oxidizing radicals generated by ozone and oxygen are highly toxic to anaerobic and aerobic bacteria depending on their concentration. For example, carbon dioxide is effective against obligate aerobes, and at high level it can prevent the growth of other microorganisms. Indirect effect: such type of effects are exerted by altering the ecology of microbial environment in which gas composition is modified. For example, replacement of oxygen by nitrogen. As soon as the surrounding atmosphere is changed, the competitive environment is also modified. Thus, atmosphere that have a negative effect on the growth of one particular microorganism may promote the growth of another microorganism, which is called positive effect. This effect may have positive or negative consequences depending upon the native pathogenic microflora and their types and the nature of the substrate. Several newer technologies are in practice to inhibit the growth of microorganisms, and most of these are based on temperature to enhance the inhibitory effects. it has been noted that controlled atmosphere and modified atmosphere packaging of certain food which can significantly enhance their shelf life. Normally, the inhibitory effect of carbon dioxide increase with decreasing temperature due to the increased solubility of carbon dioxide at a lower temperature. The pH of food becomes lower after the dissolution of carbon dioxide. In general, nitrogen, being an inert gas, mainly used to displace oxygen in food package either alone or in combination with other gases like carbon dioxide, hence showing an indirect inhibitory effect on the growth of aerobic microorganisms. Some microorganisms produce secondary metabolites such as antibiotics, bacteriocins, hydrogen peroxide and organic acids, which are either inhibitory or lethal to other microorganisms. Nowadays, the cidal or killing effect of some food microflora on others is well known. Substances produced by one microorganism either kills or inhibits the other organisms is an example of microbial interference or microbial antagonism.
Many scientists studied microbial interference. Sir Howard Florey, who gave some examples of interference back to Pasteur and Joubert in 1877, before the 1960s, such type of work has been done by clinical research. Later on, in the same 1960s, some food microbiologists showed their interest in this work. Dack and Liptsch noted that the natural flora of frozen pork pies kills inoculated cells of Staphylococcus aureus, E. coli and Salmonella typhimurium. The microbial interference are of three types: competitive exclusion, replacement therapy and lactic antagonism.
The potential of microbial growth in food is determined by the food environment and the environment where food is stored. Intrinsic factors are the parameters related to the food itself, such as nutrient, water activity, pH, redox potential and oxygen, while extrinsic factors are the environmental conditions where food is kept, such as storage temperature, relative humidity, gaseous environment, and the activity of other nearby microorganisms plays a significant role in the multiplication of microbial cells.